He, along with Dr. Paul Webster and Dr. Frank Brooks, co-founded APA 53 years back. In honor of our co-founders, we have a special event every day of the meeting. We have a Dr. Go Lifetime Achievement Award, which will be given tonight during our dinner. Then we have Paul Webster, a lecture, which will be given tomorrow. But today, we have a privilege of having Dr. Frank Brooks lecture. Frank Brooks was a gastroenterologist, one of the best and most eminent in his time. He was a chair of GI in Philadelphia, in UPenn, and he was passionate about pancreatitis and American Pancreatic Association, co-founder, president of APA. So in honor, we have a annual lecture, and this year it's my privilege to introduce our speaker, Dr. Anna Gokwaskia. Anna, my close friend for almost last 30 years, Anna, now, right? I think, and so those of you, I'm sure everyone knows, Anna is professor in Department of Medicine, UCLA, and a distinguished senior research career scientist in VA hospital in Los Angeles. Pretty soon you'll know from her accent that she was born and raised and got her PhD from Russia, the ex-Soviet Union in her time. But in 1991, she moved to Toronto, the Sick Children Hospital, one of the best in Canada. So, but pretty soon she saw the light and she wanted to be a pancreatologist, and she realized that to be an excellent pancreatologist, she needs to be in US. So she joined and collaborated for many, many years with Dr. Steve Pandol in San Diego and in Los Angeles. Anna has continuous external funding since 1993, just a year after she joined, and that has still continued. She has done almost anything and everything you can think about in experimental pancreatitis research, whether it's trypsin activation, role of calcium in acinar cells, inflammation, guru of NF-kappa B research, and many more areas which I don't have time to mention. She has almost 200 publications, very high impact publications. And for her funding, I think one of the more important thing is she was PI of first and only PO1 in the country so far. I know Dr. Serrano is standing back there. I hope that we will have more of PO1s for experimental pancreatitis. But Anna opened the path for us in that way. So she is a path breaker. So she has really amazing work in autophagy and every area. So it's really a privilege and honor. Before I forget, Anna is also remember we mentioned Dr. Gold, so she was our Lifetime Achievement Awardee last year. So welcome, Anna, and look forward to your talk.
Thank you very much. Thank you very much, APA, RPA organizers for this honor, and Ashok for this introduction. Okay, so in 1992, I immigrated from the former USSR to the United States and started working for Steve Pandol in his lab. So, okay, so how to do next? Can you show me? Okay, yes. Okay, started working in Steve Pendel's lab and on pancreatitis. Didn't know much about pancreatitis, but also at that time it was not known much about this disease. But by 1992, the tools which allowed mechanistic measurement of the pathogenesis of this disease have already been developed. And this is like animal models of pancreatitis, which replicate human manifestation of this disease have been developed, and methods to isolate functional acinar cells have been established. The mechanisms of inflammation and necrosis, which leads to multiple organ failure and mortality, were absolutely unknown. Little was known. So we started working on, I have problem, I do not know how to move the slides. Can I? Yes, so what should I do? Okay, so we started really, the first question was on what is the mechanism of the inflammatory response of pancreatitis. And at that time, little was known. So what trigger inflammatory response and sources of upregulation of cytokines in blood and links between acinar cell damage and inflammatory response were unknown. We had this idea that inflammation is initiated by injured acinar cells, that injured acinar cells express, produce and release cytokines which cause inflammation. But at this time, it was very difficult to sell because general belief was that only immune cells can release cytokines, can produce cytokines. So I remember we submitted paper to JCI in 97, and the concern of reviewers was that it is contamination with immune cells in our preparation, but not really acinar cells which produce cytokines. So we perform additional experiments and eventually convinced reviewers that it is indeed acinar cells which can express, produce, and release tumor necrosis factors which attract inflammatory cells to the area. Later, Ilya showed on isolated single acinar cells that really acinar cells express a plethora of various cytokines, not only tumor necrosis factor, and again, later we showed that acinar cells, when treated with pancreatitis inducer, express adhesion molecules, which really help to attach neutrophils to acinar cells. So really, this pathway, our data really support the pathway that injured acinar cells cause cytokine and chemokine release, which attract inflammatory cells. But the question was really, what is that in injured acinar cells which causes cytokine expression? And at that time, 1986, nuclear factor kappa-B was discovered, and also it was not well known that it is the most potent pro-inflammatory transcription factor. There appeared some publication which suggest its role in inflammation, and we decided to look at the role of NF-kappa-B in pancreatitis. So Ilya really set up the technique, electromobility shift assay, and we showed that NF-kappa-B is activated very early and robustly in cerulean pancreatitis. And also the same activation is in acinar cells. So it is a result of effect of pancreatitis inducer on acinar cells. So really, this, we spent several years then to show that it is a common event in various dissimilar models of pancreatitis, and shows that NF-κB indeed mediate production of cytokines by acinar cells. So all this confirm the idea that initial acinar cells damage by pancreatitis inducer causes NF-κB activation, resulting in cytokines release and production, and really eventually attract inflammatory cells which produce more cytokines, resulting in cytokine storm. So this data really were first which link acinar cell damage and inflammation in pancreatitis, and also the first to show that NF-κB is activated and involved in inflammatory response development in pancreatitis. So this part was clear. The second part is really these inflammatory cells which infiltrate pancreas. How do they affect acinar cells? And here we use very rough approach. What we did at that time, yes, it is 98 or so. So we injected mice and rats with anti-neutrophil serum to deplete completely neutrophils and to see the effect of this lack of neutrophils on the severity of pancreatitis. And it appears that really we have much less severe pancreatitis in animals depleted from neutrophils. And what was really unexpected, that not only necrosis decrease and serum amylase, but also trypsinogen activation markedly reduced with the neutrophil depletion. And it was again very difficult to sell and to publish. Finally, we published this in Gastroenterology, and Michael Steer, who was best expert on trypsinogen activation at that time, wrote editorial which says that this data are against the paradigm that really what we find that inflammatory cells really affect trypsinogen activation in acinar cell is against that paradigm. But we were lucky, this data were confirmed, and at least in several very elegant studies from Marcus Lerch and Julia Meyerly lab, and by other people, and it all came out to the conclusion that there is a vicious cycle between acinar cell damage and inflammation. So acinar cell damage causes release from acinar cells of cytokines through NF-κB dependent mechanism. This results in attraction on inflammatory cells. Inflammatory cells perpetuate acinar cell damage and further. And this is the initial events in pancreatitis. And I want to show you this schematic. It is from much later year. It is, but also not today, from our review with Aida and Hannah Albul, which showed how inflammatory infiltration schematic, its mechanism looks now. And it is, yes, much more mechanistically deeper than what we had, and you have additional pathway mediated by necrosis and inflammasome formation. But in the center, anyway, the same idea that acinar cells really activate pro-inflammatory transcription factor, resulting in expression, production and release of cytokines, and this is attract inflammatory cells, initiating inflammation. And really this inflammatory cells further damage acinar cells through various mechanisms. So the next area of our research during these years was mechanisms of acinar cell death in pancreatitis and role of mitochondrial dysfunction in pancreatitis. So it is well known that necrotizing pancreatitis is associated with high morbidity and mortality, but by 1992, we knew very little about the types of cell death in pancreatitis, about the underlying mechanism, and possible approaches to reduce necrosis in pancreatitis. So I just want to remind you that there are in eukaryotic cells, there are two major types of cell death, which are different in morphologic characteristics, underlying mechanism, and consequences for the organ and the whole organism. So necrosis causes loss of membrane integrity, including plasma membrane integrity, resulting in release of cellular constituents outside, and it causes inflammation, which is bad, very bad for organ and organism. Very different. Apoptosis manifests by nuclear cell shrinkage, division into apoptotic bodies, which are engulfed by macrophages, no inflammation, no release, anything outside, no inflammation, very decent cell death. 1990-2000 can be called a decade of major discoveries in cell death research. And on the top are the guys who got Nobel Prize for discovery of gene-mediating apoptosis. It's really for gene-mediating caspases, cysteine proteases, which cause apoptosis execution. And on the low row, I put photos of the guys who establish a central role of mitochondria in mediating cell death. And this is Donk, which showed critical role of cytochrome C in the apoptosis, Stanley Korsmeyer, unfortunately late, who established the role of BCL2 proteins, mitochondria proteins, in regulating both apoptosis and necrosis, and Bernardi, who showed a role of mitochondrial permeability transition PO in regulating necrosis. So we started analyzing acinar cell death in pancreatitis by measuring necrosis and apoptosis morphologically. So electron microscopy of acinar cells shows that these both type of cell death are present in pancreatitis. And you see key apoptosis characteristics, condense chromatin in nuclear apoptosis and release everything outside in necrosis. So interestingly, when we started measuring this type of cell death, it appeared that in all experimental models of pancreatitis, necrosis rate is much greater than apoptosis. In mild disease, the ratio necrosis versus apoptosis around 2-3. In more severe pancreatitis can be up to 300. And also you can see that increase in necrosis resulted in simultaneously decrease in apoptosis. So there is an inverse correlation between necrosis in apoptosis in pancreatitis. So because necrosis is a predominant type of cell death in pancreatitis, we look at the role of permeability transition PO. And this is a non-selective channel which cross mitochondria both inner and outer membrane, and in physiologic conditions it is closed. But under the action of stresses, and the most potent is calcium, so mitochondria calcium overload, this results in opening of these channels. Channels are not selective, so all molecules, including water, enter mitochondria, and it blocks mitochondria potential, mitochondria ability to synthesize ATP, and cause necrosis. Also, the composition of MPTP is not very well established. It is well established that cyclophilin D, mitochondrial resident, is a major mediator and regulator of these channels. And cyclophilin D genetic ablation really prevent MPTP opening. So here we decided to look at the role of MPTP in regulating cell death in pancreatitis. And here I show the example for ex vivo pancreatitis induced by CCK. You see that CCK causes rapid depolarization, and the same depolarization is a common event in various dissimilar models pancreatitis. This depolarization is completely prevented in mice with cyclophilin D ablation. The same ATP production is restored in these mice in pancreatitis, and necrosis goes down. And we showed that this occurs in various dissimilar models of pancreatitis. So the conclusion is that persistent MPTP opening is a common event in pancreatitis, and it mediates necrosis, which is a major type of cell death in experimental pancreatitis. By the way, apoptosis is also very, very low in human disease. So it was interesting that cyclophilin D ablation not only decrease necrosis, but also reduce severity in various dissimilar models of pancreatitis. We do not understand yet well underlying mechanism, but this data indicate a critical role of mitochondrial dysfunction in pancreatitis development. And this study were done in collaboration with Robert Sutton Lab. So what about apoptosis? What is the mechanism of apoptosis in pancreatitis? It is absolutely different from that of necrosis. Calcium does not regulate apoptosis in pancreatitis, and permeability transition pore is not involved. So the central event in apoptosis pathway is release of mitochondria resident cytochrome C in cytosols. Once in cytosols, it activates caspases, resulting in execution of apoptosis. And cytochrome C release is mediated by specific channel is in outer mitochondria membrane, does not touch inner with potential. And these channels are formed by pro-apoptotic BCL2 proteins, BAX and BAC. Channel cause MAC, mitochondria apoptosis channels. So in elegant studies from Ashok Saluja lab and Marcus Lerch lab, they showed that initial event in apoptosis pathway in pancreatitis is lysosomal permeabilization, which results in release in cathepsin B, which really proteolytically process BAX, resulting in activation of MAC channel, cytochrome C release in apoptosis. So why really the extent of cytochrome C release and apoptosis is so low in pancreatitis? So we showed that the reason is peculiar characteristic of MAC in pancreas. So in all other organs, MAC does not depend in potential differently. In pancreas, depolarization blocks cytochrome C release, block MAC opening. And this results in decrease in apoptosis pathway. So keep also in mind that depolarization occurs very rapidly. For example, in response to increase in calcium, second after there's increase in calcium, loss of potential. So, and this really blocks cytochrome C release. Further, depolarization caused decrease in ATP production, and it blocked caspase activation, which is ATP dependent. So you see that really apoptosis here does not have much chance to become a predominant pathway of death. And so unfortunately, necrosis, which is really damaging predominant type of cell death in pancreatitis. So conclusion here that parenchymal cell death in acute pancreatitis is mediated by mitochondrial dysfunction in acinar cells, and the major type of parenchymal cell death is necrosis mediated by opening of permeability transition pores. So pharmacologic approaches to inhibit PTP or cyclophilin D are promising in treatment of pancreatitis, should be promising. So what I talk is that both inflammatory pathway, cell death pathway are initiated by acinar cell damage. So what is the nature of this acinar cell damage? So in the last part of my talk, I will be discussing the mechanism of acinar cell damage, which leads to pancreatitis, in particular role of lysosomal autophagy pathway. So autophagy is a process which cells use to eliminate damaged organelles, damaged mitochondria, ER, endosome, zymogen granule, lysosome, whatever. The only one which really can do it. And here are photos of two people. First is Christian Didiou, who discovered lysosome. He was also the first to call autophagy, give this name, autophagy. And the second one is Japanese scientist who got this Nobel Prize relatively recently, 2016, for discovery of ATG proteins which mediate autophagosome formation. So major type of autophagy, macroautophagy, initiated by formation of autophagosome, which sequester cargo destined for degradation, deliver it to lysosome, where autophagosome fuse with the lysosome, forming autolysosome, where cargo is degraded by lysosomal hydrolases. When you look at the images, electron microscopic images of acinar cells from human or experimental pancreatitis, first thing you see is vacuoles, multiple vacuoles. And these vacuoles carry marker of autophagy. So they are autophagic vacuoles. And appearance of these multiple abnormal autophagic vacuoles, they have property of autolysosome because they have inside partially degraded cargo, indicates that autophagy is impaired in pancreatitis and that this impairment is at the level of lysosome. Why is it? Okay, you see on the top panel that normal efficient autophagy are very rapid, that turn over from sequester of organelle to complete degradation, elimination of vacuoles is several minutes. And also, for example, pancreas, normal human or mouse have the highest level of autophagic rate among all the organs, but we do not see these vacuoles in normal pancreas just because autophagy is very efficient. Accumulation of autolysosome indicates that something is wrong with degradation in lysosome. By some reason, it is not efficient or delayed. We further showed that really autophagic degradation is, autophagic degradation is reduced in pancreatitis much lower than in control cells or cells with starvation. And this degradation is associated with accumulation of autophagic vacuoles that should increase in the marker LC3. And this all indicates that something is wrong with lysosomal degradation in pancreatitis. It appears that pancreatitis caused multiple defects in lysosome. The first one were uncovered many years ago by Ashok Saluja and Michael Steer, which showed that lysosomal hydrolase activity is inhibited in pancreatitis. The catepsins redistributed in a different heavier subcellular fraction. And later we showed that catepsins are under process in pancreatitis, but that's not all. Pancreatitis causes significant loss in acinar cells of the major lysosomal protein, LAMP1 and LAMP2, which mediates integrity of lysosome and also proteolytic activity. Pancreatitis causes marked decrease in transcription factor EB, which mediates lysosomal biogenesis and autophagy, and also in the protein GNPTB, which mediates delivery of hydrolases to lysosome. So what all these defects are associated were observed both in human and experimental pancreatitis. So what is the functional meaning of this defect? During the last years, there have been developed 7, I think, genetic models, which specifically inhibit different steps in autophagy in pancreas. And these are ATG7 knockout, ATG5, and BMP1, which block autophagosome formation, LAMP2 GNPTB knockout, which blocks lysosomal proteolytic activity, and catepsin B and D knockout as well, and TFB, TFE3 knockout, which blocks autophagy at transcriptional level. All these genetic defects cause development of spontaneous pancreatitis, all 7 of them. And also keep in mind that this defect, many of them are observed in both experimental and human pancreatitis. This data indicates that lysosomal autophagy pathway is essential for acinar cells homeostasis, and also that impairment or blockade of this pathway initiates and drives pancreatitis. What is the underlying mechanism? And that's how we see it. Acinar cells is a factory which worked all the time, synthesize protein, process, fold, deliver to Golgi, then pack in secretory granules, purify these granules, condense them, deliver to plasma membrane. All this require tools, and tools are in the plasmic reticulum, mitochondria, and the lysosome. And these tools, because they are overused by acinar cell, very often are broken and require elimination and be replaced by new one. And in the absence of autophagy, it does not occur. So cells are overwhelmed by damaged organelle, which often initiate, like damaged mitochondrial, which I discussed above, initiate pro-inflammatory and cell death pathway. So we think that that's idea why pancreas is so sensitive to autophagy inhibition, because it is much more sensitive compared to other organs. Autophagy blockade in other organs do not cause such detrimental effect. So this is just more in detail the same. So you see ATG7 knockout, which it is standard chronic pancreatitis with all these defects, inflammatory infiltration, fibrosis, pancreatitis, histology, but also it is huge ER damage, which develops prior to this. Next is GNPTB pancreatitis, the same thing, increase in necrosis, inflammation, fibrosis associated with huge mitochondria damage caused by blockage of oxidative phosphorylation, ATP synthesis, activation of mitochondria fragmentation. Interesting that autophagy also play a key role in protection pancreas against trypsinogen activation. So, and you can see here that in all models of genetic models which block autophagy, you see significant increase in active trypsin in acinar cells. And this is likely because that with normal acinar cell functioning, there's some accumulation of trypsin in some, how Ashok says, localized organelles. Cells see this organelle as sick and they are eliminated by autophagy. But if autophagy is not working, we see this accumulation of trypsin. And in pancreatitis, where lysosomes are sick, we have accumulation of trypsin in autolysosomes. It was all genetic models. What about pancreatitis? The idea is that if we increase autophagy efficiency in pancreatitis, we will see improvement of the disease. And yes, it is a fact that TFEB activator trihalose, which improve autophagy efficiency, it also causes significant decrease in mitochondrial damage, prevent ER stress response, prevent pro-inflammatory activation of pro-inflammatory signaling, and trypsinogen activation in pancreatitis. This trihalose now is undergoing various clinical trials to treat various diseases. It is safe and would be wise to test it in repurpose it for treatment of pancreatitis. And this is the last effect, but not the least important effect of lysosomal damage on pancreatitis. And really, lysosome play a key role in cholesterol trafficking in eukaryotic cells. So on the first panel, you see marker of free cholesterol, Philippine, and you see that acinar cells is really filled with cholesterol. And if you compare level of free cholesterol in acinar cells and hepatocytes, you will see that, for example, triacylglyceride, their level is 30 times less in hepatocytes versus acinar cells, but cholesterol is exactly the same. So why acinar cells need so much cholesterol? Because, as you know, cholesterol is enriched in plasma membrane to maintain receptor conformation, ion conformation, etc. But zymogen granules membrane have exactly the same lipid composition as plasma membrane to allow fusion between zymogen granules and plasma membrane. And you can see that indeed cholesterol is fully colocalized with amylase. So it is in zymogen granules, it is also in Golgi, which forms secretory granules. So all papers showed that if you artificially reduce level of cholesterol in zymogen granules, cells die and also start triggering pro-inflammatory pathway. So what happens in pancreatitis? Cells really acquire cholesterol from the circulation, process it through endocytosis, process in lysosome, and then free cholesterol egress outside and redistributed between different organelles. In acinar cells, because of lysosomal dysfunction, cholesterol got stuck in lysosome, and you see it by increased colocalization of free cholesterol with marker of lysosomes, and this results in decreased cholesterol in plasma membrane. As a result, cell starts panic that it does not have enough cholesterol for cell needs. And there is significant activation of cholesterol synthesis pathway, which really occur both in experimental and human pancreatitis. This does not help, but results in cholesterol overload, which exacerbate mitochondria damage, lysosomal damage, ER damage, and worsen pancreatitis. Simvastatin, which inhibit cholesterol synthesis, markedly improve the severity of pancreatitis and underlying mitochondria damage. And this is the last slide, which shows how we see development of pancreatitis now see. So we think that initial event is lysosomal autophagy dysfunction, that it leads to mitochondria damage, ER stress damage, and agents which prevent it, agents that block like autophagy enhancer, that PTP inhibitors, cholesterol-lowering drug, they would improve pancreatitis. And this damage leads to activation of pro-inflammatory and pro-death pathway. And as I said, this is a vicious cycle that downstream events, inflammation and cell death perpetuate acinar cell damage. So we think that for therapeutic treatment, both acinar cell damage and inflammation and should be targeted simultaneously to improve pancreatitis. So the conclusion is that our finding showed that experimental pancreatitis and human disease strongly suggests that lysosomal autophagy dysfunction is the initial event. And now the challenge is to find what causes this event in pancreatitis. And here I want to thank all members of PRG group who worked with us for years, and our, first of all, Ilya Gukovsky and Olga Marininova, with whom we start many of this project, and Ilya really critically discussed all that, saving me from stupid directions to go. And our collaborators, and this was a joy working with them. And first of all, Stephen Pendle, with whom I started and worked for many years. Thank you.
Thank you. Thank you very much for a great oration, Anna, which covered what, 30 years of
Yes,
comprehensive review of pancreatitis research.
thank you.
Great job.
Thank you
Thank
very
you so much. Ida, would you please join us? So APA has this plaque for you to celebrate your giving a lecture, and I will have our president
Thank you very
present that
much.
to you.
Thank you. Thank you very much. Thank you very much. Thank you.
We'll break for poster review and lunch, and we'll please be a little bit late, so eat quick and spend a lot of time reviewing posters. We have some excellent posters in the other room there, and we'll see you at 2 o'clock. Thank you.